Hello, students! In this video, we are going to understand one of the most important steps of gene expression, translation. Translation is the process by which the information present in messenger RNA is used to produce a polypeptide or protein. We will begin by understanding what translation means and why it is important. Then we will examine the structure and functions of the three major types of RNA involved in protein synthesis: mRNA, rRNA, and tRNA. After that, we will follow the complete process of translation step by step, beginning with charging of tRNA, followed by initiation, elongation, and termination. Now that we know the overall pathway from DNA to protein, let us focus on the final step, translation. Translation refers to the polymerization of amino acids to form a polypeptide. The sequence of amino acids in the polypeptide is determined by the sequence of bases present in the mRNA. In simple words, the information stored in the form of mRNA codons is converted into a specific sequence of amino acids. These amino acids are joined together by peptide bonds. Translation is therefore essential because it converts genetic information into functional proteins. These proteins perform most of the important functions inside a cell. To understand how this conversion takes place, we first need to know the roles of the three major types of RNA involved in the process. Let us begin with the first type of RNA, mRNA, or messenger RNA. As its name suggests, mRNA carries the genetic information copied from DNA. This information determines the order in which amino acids will be arranged in the polypeptide. A translational unit of mRNA is flanked by a start codon, usually AUG, and a stop codon. Between these two signals, the mRNA contains codons such as GCU, AAA, and UGG, which specify the amino acids added to the growing polypeptide chain. At both ends of the mRNA, there are untranslated regions, or UTRs. These are the 5' UTR and the 3' UTR. Although these regions are not translated into protein, they help in the efficient regulation and initiation of translation. Now that we have seen the message carried by mRNA, let us look at the structure that reads this message, the ribosome. The ribosome is made up of rRNA, or ribosomal RNA, and ribosomal proteins, about 80 different proteins. Together, they form the cellular machinery responsible for protein synthesis. The ribosome consists of a small subunit and a large subunit. The small subunit interacts with the mRNA, while the large subunit contains the sites where amino acid-bearing tRNAs bind. There are two sites in the large subunit for subsequent amino acids to bind to, that is, A site or aminoacyl site, and thus be close enough to each other for the formation of a peptide bond, which takes place at P site or peptidyl site. In bacteria, 23S rRNA performs the catalytic function involved in peptide bond formation. Because it acts as an RNA catalyst, it is considered a ribozyme. So mRNA provides the message, and the ribosome provides the platform for reading that message. The molecule that brings the correct amino acids to this platform is tRNA. The third type of RNA is tRNA, or transfer RNA. tRNA is called the adapter molecule because it connects the nucleotide language of mRNA with the amino acid language of proteins. One end of the tRNA carries a specific amino acid, while another region contains the anticodon. The anticodon forms complementary base pairs with the appropriate codon on mRNA. In this way, tRNA brings the correct amino acid to the ribosome according to the information present in the mRNA. Before a tRNA can perform this function, however, its correct amino acid must first be attached to it. This brings us to the first stage of the translation process, charging of tRNA. Before translation actually begins, the amino acids must be activated and attached to their corresponding tRNAs. This process is called charging of tRNA, or aminoacylation of tRNA. The enzyme responsible for this reaction is called aminoacyl-tRNA synthetase. Each amino acid has a specific synthetase that recognizes the correct amino acid and its corresponding, or cognate, tRNA. First, the amino acid is activated using energy from ATP. The Aminoacyl-tRNA synthetase then attaches the activated amino acid to the 3' end of the appropriate tRNA. The resulting molecule is called a charged tRNA, or aminoacylation tRNA. This enzyme is highly specific, so it helps ensure that the correct amino acid is attached to the correct tRNA. Charging ensures that the correct amino acid is delivered to the ribosome during protein synthesis. Once the tRNAs are charged, they are ready to participate in the assembly of the polypeptide. The next stage is initiation.
Now that the tRNAs are charged, translation can begin with the stage called initiation. The small ribosomal subunit binds to the mRNA and moves along it until it recognizes the start codon, AUG. The initiator tRNA recognizes AUG through its complementary anticodon and brings the amino acid methionine. The large ribosomal subunit then joins the complex, forming the complete ribosome. At this point, the mRNA, ribosome, and initiator tRNA are correctly positioned, and the translation machinery is ready to add the next amino acid. This leads us to the next stage, elongation.
After initiation comes the elongation stage, during which the polypeptide chain grows one amino acid at a time. A charged tRNA enters the ribosome, and its anticodon pairs with the appropriate codon on the mRNA. The amino acid carried by this tRNA is positioned close to the growing chain. A peptide bond then forms between the amino acids. Next, the ribosome moves forward by one codon along the mRNA. The empty tRNA exits, while the next charged tRNA enters and recognizes the next codon. This cycle of codon recognition, peptide bond formation, and ribosome movement continues repeatedly. As a result, amino acids are added one by one, and the polypeptide chain continues to grow until the ribosome reaches a stop codon. After the polypeptide has grown through repeated elongation cycles, the ribosome eventually reaches a stop codon. This marks the beginning of the final stage, called termination. Stop codons do not code for an amino acid, so no tRNA pairs with them. Instead, a release factor recognizes the stop signal and binds to the ribosome. This causes the completed polypeptide chain to be released. The ribosome then dissociates into its subunits, and the mRNA is released. In this way, translation comes to an end, and the newly formed polypeptide can fold and function as a protein. Now that we have followed the complete process, let us quickly revise the 4 stages of translation. First, during charging of tRNA, each amino acid is attached to its corresponding tRNA by the enzyme Aminoacyl-tRNA synthetase using energy from ATP. This produces a charged tRNA. Second, during initiation, the ribosome recognizes the start codon, AUG, and the initiator tRNA brings methionine. Third, during elongation, charged tRNAs recognize successive codons, peptide bonds are formed, and the ribosome moves along the mRNA while the polypeptide chain grows. Finally, during termination, the ribosome reaches a stop codon. A release factor binds, the completed polypeptide is released, and the ribosome dissociates. Thus, the information present in mRNA is translated into the specific amino acid sequence of a protein. Pause the video and go through these questions for better understanding of the topic. Understanding translation becomes much easier when you remember one simple idea. mRNA provides the message, tRNA brings the amino acids, and the ribosome coordinates their assembly into a protein. The enzyme Aminoacyl-tRNA synthetase ensures that each tRNA is loaded with the correct amino acid before translation begins. Together, these components convert genetic information into a functional polypeptide. Thank you for watching. Keep learning, keep revising, and I'll see you in the next biology lesson.